All right, let's dive into a topic you guys specifically requested. Bottom-up proteomics.
Oh, this is a fun one.
I know, it's like taking a puzzle, the proteome, and breaking it down into its pieces to
Yeah.
understand the bigger picture. And
Exactly.
we've got this great excerpt from a scientific overview, but don't worry, we're
Yeah.
going to keep it nice and engaging for you. So,
Okay, so like think of it
yeah,
like taking apart a finely tuned machine, right?
yeah,
To examine each gear and spring. And so by analyzing these individual components,
right.
we can gain these insights into how the whole thing functions.
Okay, I love that. I love that analogy. So how do scientists actually do this? Like, what are the steps involved in this molecular disassembly?
Well, it all starts with extracting proteins from a sample, which could be anything from cells to tissues,
Right.
and then comes the chopping.
Okay.
So using enzymes like trypsin to break those proteins down into smaller, more manageable peptides.
So it's like trypsin acts like tiny molecular scissors.
Exactly, making precise cuts. And these cuts, what's cool is that they create peptides with consistent chemical signatures, making them easier to analyze later on.
Right.
But hold on, we're not done yet. We need to clean up the workspace, so to speak.
Right. Got to get rid of that molecular clutter.
Exactly.
So what's the next step in this protein puzzle-solving process?
So it's purification.
Okay.
Imagine trying to analyze a jumbled mess of peptides. It would be overwhelming. So purification removes any contaminants that could interfere with the analysis, like leftover chemicals from the extraction process.
Okay. So we've chopped up the proteins, we've cleaned up the mess. Now do we start analyzing these tiny pieces?
Not quite yet.
Okay.
First, we need to separate them. And this is usually done using reversed phase liquid chromatography,
Okay.
a technique that sorts peptides based on their, properties.
Wait, is this where that racetrack analogy comes in?
Yes, you got it.
Okay.
So hydrophobic peptides, those that are water avoidant, tend to cling to the stationary phase,
Yeah.
while hydrophilic ones race ahead.
Yeah.
And this separation ensures a smooth and organized flow of peptides into the next stage, which is where things get really exciting.
Okay, I'm ready. What is this exciting next stage?
It's the heart of bottom-up proteomics, mass spectrometry.
Okay.
This is where we finally identify and quantify those individual peptides. Think of it as a molecular fingerprint analysis.
Hold on. So each peptide has its own unique fingerprint. How does that work?
Each peptide has a unique mass-to-charge ratio that the mass spectrometer can actually measure.
Okay.
And then we compare these measurements to a database of known proteins,
Right.
kind of like matching fingerprints to identify a suspect.
Wow, that's incredible.
It is pretty incredible.
We're identifying these tiny protein fragments with such precision.
Right.
But you mentioned quantification too.
Yeah.
So what's that all about?
So quantification tells us how much of each protein is present. And this is crucial for understanding how protein levels change under different conditions.
Okay.
Like comparing healthy cells to diseased cells.
Oh, okay. So we're not just identifying the players, we're also trying to figure out their roles in the cellular drama.
Exactly. You got it.
That's really cool.
But here's the kicker.
Yeah.
Interpreting all this data can be a real bottleneck.
Okay.
Sometimes it takes months, even years, to fully understand the information gathered in just a week.
Wow, that's a long time. Why does it take so long to connect the dots?
Well, you think about it, we've identified thousands of proteins, right?
Yeah.
But now we need to figure out what they do, how they interact,
Right.
what their roles are in the cell. It's like having a giant list of ingredients, but no recipe.
Oh, okay. So data interpretation is like writing the recipe book for the cell.
Exactly. And it requires a deep understanding of cellular pathways, protein functions, and the specific research question being investigated.
Okay.
It's where we transform that raw data into meaningful biological insights.
All right, so we've got the basic process down. Now let's dive into some specific types of experiments that use this bottom-up approach. Our source mentioned something called discovery-based, unbiased mapping of proteins. What exactly does that mean?
Well, it's like creating a comprehensive inventory of all the proteins present in a sample
Okay.
and understanding how their abundance changes across different conditions.
Right.
For instance, comparing healthy cells to diseased cells.
So it's about getting a big picture view of the protein landscape and seeing how it shifts.
You got it.
Okay.
And to make sure this picture is accurate, scientists use multiple replicates.
Okay.
It's like repeating an experiment several times to confirm the results and to estimate the variance in protein measurement.
Hold on. 3 replicates for each condition. That seems like a lot. It's like baking a cake 3 times.
Yeah.
Just to make sure the recipe is right.
Well, not exactly, but the principle is similar. By having multiple replicates, researchers can be more confident that any observed differences in protein levels are real
Right.
and not due to random chance.
Yeah. That makes sense. All right, so we've got discovery-based proteomics down. Now let's move on to something that sounds particularly captivating, cross-linking mass spectrometry, or XLMS. Well, what's so special about this technique?
XLMS takes us beyond simply identifying proteins.
Okay.
It allows us to study how they interact with each other. It's like taking a snapshot of proteins caught in the act of interacting,
Okay.
freezing them in place so we can study their relationships.
Okay, I'm intrigued. Walk me through how this actually works.
All right, so first, we create a system where protein interactions are happening, either in a test tube or within living cells.
Okay.
Then we add a special ingredient, a cross-linking agent. Think of it as molecular handcuffs,
Okay.
linking 2 interacting proteins together.
So we're setting a trap for these interacting proteins, catching them red-handed, so to speak.
Exactly. Then, just like in the general bottom-up process, we break these proteins down into peptides.
Right.
But here's the twist. Some of those peptides will still be linked together by those molecular handcuffs, providing evidence of the original protein-protein interaction.
Wow, that's so clever. So we're using these linked peptides as clues to figure out which proteins were interacting.
Precisely. We then analyze these linked peptides using tandem mass spectrometry, which can actually pinpoint the specific amino acids involved in the cross-link.
Oh, wow.
And this information helps us create a map of protein interactions, showing which proteins are connected and where they bind to each other.
It's like piecing together a puzzle to see how the proteins fit together structurally.
Yeah.
I bet this has some amazing applications.
You bet it does. One fascinating application is protein docking, where scientists use the cross-link data to create computational models that predict the 3D structure of protein complexes. It's like being able to visualize the molecular dance between proteins in all its glory.
Wow, it's like having a front row seat to the intricate choreography of cellular life. Now, our source also mentions another set of techniques that sound equally fascinating. Proximity labeling techniques. Are these similar to XLMS in that they reveal protein interactions?
Absolutely. Proximity labeling techniques take advantage of enzymes that can tag nearby proteins. Imagine a protein holding a spray can that tags everything around it. That's the basic idea behind these techniques.
I love that analogy. So vivid. Our source really focuses on 2 main types, Apex-MS and BioID. Let's start with Apex. How does that work?
All right, so Apex-MS uses an enzyme called peroxidase, which is attached to a protein of interest.
Okay.
And when we add a specific chemical, the peroxidase goes to work, tagging nearby proteins with a molecule called biotin.
So biotin acts like a molecular beacon, highlighting those proteins that were hanging out near our protein of interest.
Precisely. And these biotin-tagged proteins can then be isolated and identified using mass spectrometry. What's really cool is that Apex can actually capture interactions happening in living cells, including those that are, fleeting and transient.
That's amazing. Are there any limitations to this technique?
Well, no technique is perfect. The source points out that Apex can sometimes lead to nonspecific labeling. Imagine our spray can wielding protein getting a bit overzealous and tagging proteins that weren't actually interacting with it.
So we might get some false positives, like mistaking a bystander for a close associate.
Exactly. And it can also be difficult to distinguish between direct and indirect interactions.
Right.
Was a tagged protein directly interacting with our protein of interest, or was it just in the vicinity?
Okay, so Apex is great for capturing those fleeting interactions, but we need to be mindful of those potential false positives. Now, what about BioID? How does that differ from Apex?
So BioID uses a different enzyme called biotin ligase to tag nearby proteins.
Okay.
It's similar to Apex in that it can capture those transient interactions.
Right.
But here's what's intriguing. BioID is actually slower than Apex.
Hold on. Slower. Isn't faster usually better in science?
Not always. Because BioID is slower, it can capture interactions that occur over longer timescales. It's like casting a wider net to catch even those fleeting encounters between proteins.
Fascinating. So it's a trade-off between speed and sensitivity.
You got it.
Apex is quick, but it might miss those slow-burning interactions. BioID takes its time, but it catches a wider range.
Exactly. And to add another layer to this, our source also mentions a faster, more commonly used version of BioID called Turboid.
Oh, okay. So now we have a whole toolkit of proximity labeling techniques to study these protein
We
interactions.
do.
But after all the excitement of protein extraction and cross-linking and proximity labeling, there's another step we can't overlook. Our source mentions peptide purification and fractionation.
Mhm.
Why is this step so important?
Remember how we talked about cleaning up the workspace earlier?
Yeah.
Well, this is where we ensure that our peptide samples are as pristine as possible before we analyze them. Imagine trying to solve a puzzle with dirt and pieces. It would be much harder.
Right. Yeah, that makes sense. We need to remove anything that might interfere with our analysis and make sure we're working with the purest possible peptide samples. But how do scientists actually achieve this level of molecular cleanliness?
There are several methods, but one of the most common is solid phase extraction, or SPE. Think of it like using a coffee filter. The filter traps the coffee grounds, unwanted molecules, allowing the delicious coffee, our peptides, to pass through.
So we're using a special filter to selectively capture the peptides we want while letting the contaminants go down the drain.
Exactly. And our source actually provides a really helpful illustration of this process, highlighting the importance of choosing the right solutions for binding, washing, and eluting the peptides. It's all about achieving this delicate balance to ensure we capture the targets we want while effectively removing those molecular contaminants.
So SPE is like a molecular sorting game, ensuring we have a clean and organized set of peptides for analysis. Now, our source also mentions other purification methods, Hillick, Ehrlich, and IEF. Are these similar to SPE or do they work in different ways?
They all serve the same purpose, purifying those peptides, but they utilize different principles and chemistries. Think of it like having different tools in a toolbox. Each tool is designed for a specific task and offers unique advantages depending on the nature of the peptides and the contaminants we're dealing with.
Okay, so it's all about choosing the right tool for the job,
Mm-hmm.
making sure we're using the most effective method to purify our precious peptide samples. But once we've got our purified peptides, what happens next? Do we finally get to analyze them?
We're getting closer. After purification comes another crucial step, liquid chromatography, or LC.
Okay.
This technique is the workhorse of proteomics. It separates those peptides based on their unique physical and chemical properties.
So it's like sorting laundry, separating the whites from the colors before throwing everything in the dryer.
That's a great way to put it. And you mentioned reversed phase liquid chromatography earlier, which happens to be the most common type used in proteomics.
Yeah, the one with the racetrack analogy.
Yes.
So how does reversed phase LC actually work?
Remember how we talked about hydrophobic and hydrophilic peptides?
Yeah.
Well, in reverse phase LC, peptides are separated based on their affinity for a hydrophobic stationary phase.
Okay.
The more hydrophobic a peptide, the longer it clings to the stationary phase, while hydrophilic peptides kind of zip through quickly.
So it's like a molecular obstacle course, with some peptides clinging to the obstacles while others navigate them with ease.
You got it. And by carefully controlling the gradient, that is, the rate at which the organic solvent concentration changes,
Okay.
we can optimize the separation of these peptides.
So we're fine-tuning the conditions to ensure those peptides are separated with the utmost precision.
Exactly.
What about those trapping and pre-columns our source mentions? What role do they play in this separation process?
Think of them as guardians protecting the analytical column and ensuring the robustness of the LCMS system.
Okay.
They act like filters, removing any remaining contaminants and preventing them from reaching the delicate mass spectrometer.
So they're like the bouncers at a molecular nightclub.
Exactly, making sure only the right peptides get in. They're essential for maintaining the purity and integrity of that separation process. And once we have our beautifully separated peptides, we're finally ready for the grand finale. Mass spectrometry.
Okay, the moment we've all been waiting for. This is where we finally get to identify and quantify those individual peptides, right?
You bet.
Yeah.
Mass spectrometry is like shining a spotlight on each peptide, revealing its unique mass-to-charge ratio. And by comparing these measurements to a vast database of known proteins, we can confidently identify each peptide and its corresponding protein.
It's like solving a molecular mystery, using the unique fingerprints of each peptide to track down its protein origin. But wait, you also mentioned tandem MS or MSMS. What's that all about? Is it like a supercharged version of mass spectrometry?
You could say that. In tandem MS, we take those identified ions and fragment them, creating a spectrum of smaller fragment ions.
So we're breaking those peptides down even further, creating even more clues to analyze.
That's a great way to think about it. And just like with purification and separation, there are various types of mass analyzers we can use,
Okay.
each with its strengths and limitations.
Our source mentions several types. TOF, quadrupole mass filter, quadrupole ion trap, FTICR, and Orbitrap. Are these like different models of mass spectrometers, each with its own unique features?
Exactly. Each type offers a different combination of sensitivity, resolution, and speed, allowing researchers to tailor their approach to the specific needs of their experiments.
So it's like choosing the right camera for a photo shoot,
A perfect analogy.
depending on whether you need high resolution, fast shutter speed, or specific lighting conditions.
Exactly. For example, our source highlights the incredible sensitivity and resolution of FTICR-MS. It's like having a high-powered microscope that can zoom in on those molecular details with incredible precision. However, this level of sophistication comes with a price.
Oh, okay.
It requires high-performance magnets and precise temperature control, making it a more complex and expensive technique.
So FTICRMS is like the high-end sports car of mass spectrometry,
Yes.
offering incredible performance, but requiring a bit more care and attention.
Exactly. But speaking of sophisticated techniques, our source introduces us to another dimension of data, ion mobility spectrometry, or IMS. What exactly is this IMS, and how does it add to the already impressive capabilities of mass spectrometry? IMS adds a whole new layer of information by separating ions based on their size, shape, and charge.
Okay.
Imagine sorting marbles by rolling them through a maze. Different shapes and sizes will navigate the maze differently.
Okay.
And IMS exploits this principle to further separate and characterize those ions.
Oh, I love that analogy. So we're essentially adding another dimension to our analysis,
Exactly.
going beyond just mass and charge to consider the physical characteristics of each ion.
Precisely. And just like with mass analyzers, there are different types of IMS techniques, each with its unique features and advantages.
Our source lists DTIMS, FAMES, TIMS, and SLIM. Are these like different maze designs for sorting those molecular marbles, each one offering a different challenge and revealing different information about the ions?
You got it. Each technique offers a different combination of resolution, speed, and sensitivity. For instance, DTIMS is known for its high resolving power, while TIMS can actually trap ions, ensuring we don't miss any clues. It's like catching and examining every single marble in the maze.
Wow, so we've got these incredible tools for separating, identifying, and characterizing peptides.
do.
But after all this high-tech analysis, we're left with a mountain of data. How do researchers make sense of it all?
That's where data analysis comes in, the final stage of our bottom-up proteomics journey.
Okay.
This is where we transform raw data into meaningful biological insights, connecting the identified proteins to the research question at hand. And trust me, it's a journey in itself.
All right, well, I'm ready to embark on this data analysis adventure. Let's dive in and see how researchers make sense of this molecular treasure trove.
Data analysis is where we turn that mountain of data into something we can actually understand and use. It's like having a room full of puzzle pieces. We need to sort them, assemble them, and figure out what the picture tells us.
Okay, so it's not just about identifying the pieces, it's about understanding the bigger picture they create. What are some of the key steps involved in this data analysis puzzle-solving process?
First, we need the right tools. There are several software programs designed specifically for analyzing proteomics data, each with its own strengths.
Our source mentions quite a few: MaxQuant, MSFragger, Mascot, MSGF+, Penn, XTandem, and Comet. Are these like different types of puzzle-solving software, each offering a different approach to analyze the data?
Exactly. Some are better at identifying peptides, while others excel at quantification or analyzing specific types of data, like that cross-linking data from XLMS.
Speaking of cross-linking data, our source highlights one software called Kojak. What's so special about Kojak? Does it have some unique puzzle-solving skills?
What's fascinating about Kojak is that it's specifically designed to analyze data from cross-linking experiments, helping researchers untangle those complex protein relationships. It's like having a detective who specializes in solving molecular mysteries.
Okay, so Kojak is our go-to detective for figuring out those protein partnerships. But regardless of which software we use, there are some fundamental steps involved in data analysis, right? Our source mentions quality control is a crucial step. Why is that so important?
Quality control is like double-checking your work to make sure everything is accurate and reliable. We need to identify and address any issues or inconsistencies in the data before moving on to further analysis. It's like making sure all the puzzle pieces are the right size and shape before trying to fit them together.
So it's all about ensuring the integrity of the data, making sure we're not working with faulty pieces that could lead to a distorted picture.
Exactly. And then there's data normalization. Think of it like leveling the playing field so we can accurately compare data from different samples or experiments.
Okay, so normalization is like adjusting for different lighting conditions in a photograph, making sure we're comparing apples to apples.
That's a great way to think about it. And there are various normalization methods we can use, each with its own strengths and limitations, depending on the type of data we're working with.
So it's about choosing the right tool for the job, ensuring we're using the most appropriate method to normalize the data and avoid any misleading conclusions.
Precisely. And then there's batch correction, which is particularly important for large studies where samples are processed in multiple batches. It's like removing unwanted noise from a signal, ensuring that any observed differences are due to biological factors and not technical artifacts.
So batch correction is like filtering out static from a radio signal, making sure we're hearing the true message loud and clear.
A perfect analogy. And once we've gone through all these steps, quality control, normalization, and batch correction, we're finally ready for the most challenging and rewarding part of the journey: data interpretation.
Ah, the moment we've all been waiting for. This is where we finally get to connect the dots, transforming raw data into meaningful biological insights. But as you said, it's also the most challenging part. Why is that? What makes data interpretation so tricky?
Well, we've identified thousands of proteins and their interactions, but now we need to figure out what it all means. What are these proteins doing? How do they work together? What roles do they play in the cell? It's like having a giant jigsaw puzzle completed, but not understanding the picture it depicts.
So we've assembled the puzzle, but now we need to decipher its message, figure out the story it's telling us.
Exactly. And this requires a deep understanding of cellular processes, protein functions, and the specific research question we're trying to answer.
It's like being a detective who's gathered all the evidence but still needs to crack the case, figure out who did it and why.
Precisely. And to help us with this detective work, our source mentions several approaches, like protein networks and functional enrichment analysis.
Okay, let's unpack those. What are protein networks, and how do they help us make sense of this data?
So protein networks help us visualize the relationships between proteins. They show which proteins interact and how those interactions might influence cellular pathways. It's like mapping out the social network of a cell, seeing who's connected to whom and how information flows through those connections.
So instead of just seeing a list of names, we're seeing a web of connections, understanding how those proteins are working together as a community.
Exactly. And then there's functional enrichment analysis, which helps us understand what biological processes those proteins are involved in. It's like taking a group of people and figuring out what they have in common. Are they all musicians, athletes, or perhaps scientists? In the case of proteins, we're looking for shared functions or pathways that might provide clues to their roles in the cell.
So we're grouping proteins based on their shared interests or skills, getting a better understanding of their potential roles in the cellular orchestra.
Precisely. And all of this analysis, from identifying peptides to understanding their functions, relies heavily on those protein sequence databases we discussed earlier. They're the Rosetta Stone that helps us translate the mass spectrometry data into meaningful biological information.
Speaking of protein sequence databases, our source delves into some nuances about choosing the right database for a specific research question. They emphasize that it's not a one-size-fits-all situation.
That's right. Selecting a database that's too large can lead to false positives. Those pesky decoy hits that make us think we found something significant when we haven't. On the other hand, a database that's too small might cause us to miss important proteins altogether, increasing false negatives.
So it's like trying to find a needle in a haystack. If the haystack is too big, we might mistake a piece of straw for the needle, but if the haystack is too small, we might miss the needle entirely.
A perfect analogy. The source suggests considering several factors when choosing a database, such as the specific organism being studied, the need to differentiate between protein isoforms, and the capabilities of the search algorithm being used.
They also highlight a challenge that arises when combining data from multiple studies. Each study might have used a different database and different search parameters, making it tricky to compare the results directly.
That's where PeptiAtlas comes in. It addresses this challenge by collecting data from numerous public studies, reanalyzing it using a consistent pipeline, and controlling for false discoveries at the whole atlas level.
So it's like a meta-analysis of proteomics data, bringing together information from diverse sources to create a more comprehensive and reliable picture.
Precisely. And beyond just identification, Peptide Atlas also provides information about peptide detectability, which can be helpful for designing targeted proteomics experiments. It's like having a roadmap that guides researchers toward the most promising avenues for discovery.
Okay, so Peptide Atlas is a powerful tool for navigating the vast sea of proteomics data, ensuring that we're working with the most reliable and comprehensive information available. But once we've identified those proteins and their interactions, the next big question is, what do they do? How do we validate their functions?
That's a great question. And our source highlights the incredible power of CRISPR-Cas9 gene editing technology for precisely this purpose. It's revolutionized our ability to manipulate genes, enabling us to target specific proteins and study the effects of their depletion or overexpression.
So CRISPR-Cas9 is like having a molecular scalpel, allowing us to surgically remove or enhance the activity of a specific protein in the cell and see what happens.
You got it. By observing the consequences of these manipulations, we can gain valuable insights into the function of the targeted protein and its role in cellular processes.
That's amazing. It's like being able to tweak the cellular machinery with incredible precision and see how those tweaks affect the overall system. But are there any potential pitfalls with using such a powerful tool?
Well, as with any powerful tool, there are considerations. Our source cautions that CRISPR-Cas9 can sometimes have off-target effects, meaning it might unintentionally edit genes other than the intended target.
So it's like accidentally hitting the wrong key on a piano, creating an unintended note. We need to be careful and ensure that our edits are precise and targeted.
Exactly. And they also point out that long-term depletion or overexpression of a protein can sometimes lead to compensatory mechanisms in the cell, making it difficult to interpret the results.
So the cell might try to compensate for the missing or overabundant protein, throwing a wrench into our experimental observations.
That's right. To address these concerns, researchers often use alternative gene manipulation techniques, such as siRNA-mediated knockdown or degron-based degradation systems. These techniques provide more transient control over protein levels, reducing the risk of compensatory effects.
So it's like having a dimmer switch instead of an on-off switch, allowing for more fine-tuned control over protein levels.
Precisely. And our source also emphasizes the importance of using appropriate controls in these experiments. For instance, expressing the CRISPR-Cas9 machinery without a guide RNA can help assess the baseline effects of the editing system itself. It's like having a placebo group in a clinical trial, allowing us to distinguish the true effects of our manipulation from any background noise.
Okay, so CRISPR-Cas9 is a powerful tool for validating protein function, but we need to use it wisely and interpret the results carefully. Now, in addition to understanding protein function, we're also interested in understanding where those proteins are located within the cell. How do researchers study protein localization?
There are several techniques, each offering a unique perspective on protein distribution within the cell. Immunofluorescence microscopy, GFP tagging, and subcellular fractionation are a few examples mentioned in our source.
Okay, let's break those down. What's immunofluorescence microscopy all about? How does it help us visualize protein location?
Immunofluorescence microscopy uses antibodies tagged with fluorescent dyes to highlight specific proteins within cells or tissues. It's like shining a spotlight on the protein of interest, allowing us to see where it resides in the cell.
So we're using glowing antibodies as molecular detectives to track down our protein of interest and reveal its hiding spot within the cell.
You got it. And then there's GFP tagging, which involves attaching green fluorescent protein, GFP, to the protein of interest. This allows us to track its movement and localization in living cells.
So it's like giving the protein a glowing beacon that reveals its whereabouts in real time.
Exactly. We can actually watch the protein move around the cell and see where it goes and what it interacts with. And then there's subcellular fractionation, which involves separating different cellular compartments like the nucleus, cytoplasm, and mitochondria,
Right.
and then analyzing the protein content of each fraction.
So it's like sorting laundry into different baskets based on color or fabric type, allowing us to see which proteins belong to which cellular compartment.
A perfect analogy. This technique provides insights into the distribution of proteins across different cellular neighborhoods, helping us understand how proteins are organized and trafficked within the cell.
All right, so we've got a whole toolkit of techniques for visualizing and understanding protein localization. But how does all of this fit into the bigger picture of data interpretation? How do we connect the dots between protein identification, function, and localization to create a cohesive understanding of the cell?
That's a great question, and it brings us to a crucial point, the integration of proteomics data with other types of biological data. Our source emphasizes the importance of combining proteomics with genomics, transcriptomics, and metabolomics to create a more holistic understanding of biological systems.
Okay, so it's not just about studying proteins in isolation. It's about understanding how they fit into the broader context of the cell and the organism as a whole.
Exactly. Genomics provides information about the DNA sequence, transcriptomics tells us about gene expression, and metabolomics reveals the metabolic profiles of cells or organisms. By integrating these different layers of information, we can gain a much deeper understanding of how genes, proteins, and metabolites interact to regulate cellular processes and contribute to health and disease.
It's like having different pieces of a puzzle that, when combined, reveal a more complete and detailed picture of the overall system.
Precisely. And with the increasing availability of large-scale data sets and sophisticated computational tools, the field of integrative biology is rapidly advancing, enabling researchers to make connections and draw conclusions that were previously impossible.
This is all incredibly fascinating, but it also sounds incredibly complex. How do researchers even begin to make sense of such a vast and intricate web of data?
It's definitely a challenge, but the rewards are immense. By combining the power of bottom-up proteomics with other omics approaches and sophisticated computational tools, researchers are gaining unprecedented insights into the molecular mechanisms that govern life. And with every new discovery, we're getting closer to unlocking the secrets of health and disease.
It really is mind-blowing to think about all these intricate processes happening within our cells and how scientists are using these incredible tools to piece together the puzzle of life. But with so much information and so many techniques, it's easy to get lost in the details. What are some of the key takeaways about bottom-up proteomics that our listeners should remember?
Well, I think one of the most important takeaways is the sheer power of this approach. By breaking down proteins into their individual peptide components, we're gaining an incredibly detailed view of the proteome, the complete set of proteins within a cell or organism.
It's like taking inventory of a vast library, cataloging each book and its contents to understand the breadth and depth of knowledge contained within.
Exactly. And this knowledge is crucial for understanding virtually every aspect of biology, from the basic mechanisms of cellular function to the complex processes that drive health and disease.
We've talked about identifying proteins, quantifying them, studying their interactions, and even manipulating their function. It's amazing to think about how far this field has come and the incredible insights it's providing.
Absolutely. And as technology continues to advance, we can expect even more sensitive and powerful tools, allowing us to probe deeper into the proteome and uncover new layers of complexity.
It's exciting to think about the discoveries that await us. What are some of the potential future applications of bottom-up proteomics that you're particularly excited about?
Well, one area that holds immense promise is personalized medicine. Imagine being able to create a detailed proteomic profile for each individual, providing insights into their unique health risks and guiding tailored treatment strategies.
So instead of a one-size-fits-all approach to medicine, we could have treatments that are specifically designed for an individual's unique proteomic makeup. That would be revolutionary.
It would, indeed. And it's not just about treatment. Proteomics could also play a crucial role in early disease detection. By identifying specific protein biomarkers that are associated with certain diseases, we could develop diagnostic tests that can detect those diseases at their earliest stages when treatment is most effective.
It's like having a molecular early warning system, alerting us to potential health problems before they even manifest as symptoms.
Exactly. And with ongoing advancements in technology, the possibilities for future discoveries are truly limitless. We might even be able to map the entire human proteome in exquisite detail, understanding the function of every single protein and how they interact to create the symphony of life.
Wow, that's a mind-boggling thought. It's like having a complete blueprint of the human body at the molecular level, revealing all the intricate connections and interactions that make life possible.
And thanks to resources like Peptid Atlas, which we discussed earlier, researchers around the world can share their data and collaborate to accelerate these discoveries. It's a testament to the power of open science and the collective pursuit of knowledge.
Absolutely. By working together and sharing our knowledge, we can unlock the full potential of bottom-up proteomics and revolutionize our understanding of biology and human health. But even with all this incredible technology and collaborative effort, there's still so much we don't know about the proteome.
That's right. We're only scratching the surface of what we can learn. For example, we still don't fully understand how proteins fold into their unique 3D structures, which is crucial for their function. And we're only beginning to unravel the complexity of protein interactions and how they regulate cellular processes.
So even with all the amazing progress that's been made, there's still a sense of mystery and wonder surrounding the world of proteins.
Absolutely. And that's what makes this field so exciting. With every new discovery, we're opening up new avenues for exploration and uncovering new layers of complexity.
It's like exploring a vast and uncharted territory, where every step reveals something new and unexpected. So as we wrap up this deep dive into bottom-up proteomics, what's the one thing you'd like our listeners to take away from this episode?
I'd say the most important thing to remember is that we're living in an era of incredible scientific discovery. Thanks to powerful tools like bottom-up proteomics, we're gaining an unprecedented understanding of the molecular mechanisms that govern life.
And these discoveries have the potential to revolutionize everything from medicine to agriculture to our understanding of the natural world.
Absolutely. So I encourage everyone to stay curious, keep asking questions, and explore the fascinating world of proteins. You never know what amazing discoveries you might uncover.
Well said. And for our listeners who want to delve deeper into this fascinating field, we highly recommend checking out resources like Pepti Atlas and exploring the wealth of information available online.
There's a whole universe of knowledge out there waiting to be discovered, and bottom-up proteomics is one of the most powerful tools we have for unlocking its secrets.
Thank you so much for joining us on this deep dive into the incredible world of bottom-up proteomics. We hope you've enjoyed the journey and learned something new along the way.
It's been a pleasure sharing my knowledge with you and your listeners.
And until next time, keep those questions coming, and we'll see you on our next deep dive.